Welcome to MLT 130, Clinical Chemistry. The first unit and lecture that we're going to be covering is carbohydrates. Here are the objectives for this unit. You can pause the video at this time and read through them on your own. Our body uses carbohydrates for energy and structural components such as glycoproteins and glycolipids. Our brain and red blood cells and retinal cells require carbohydrates as our main source of energy. And our retinal cells are the cells in the retina of our eye. The carbs are the main energy supply for the body, and they're stored in the glycogen in the liver and the muscle. About 50 to 90% of carbs are taken in the forms of grain, starchy vegetables, and legumes. And legumes are things like peas, beans, peanuts, and lentils. Excess carbs are also stored as adipose tissue, or also known as fat. Now let's talk about the classification of our carbohydrates. The first two classifications that we need to discuss is monosaccharide and disaccharide. The monosaccharide is also referred to as a monomer or a simple structure. The number of carbons is used to help categorize these simple sugars, and here is the formula for our monosaccharides. The simplest monosaccharides are those with only 3 carbon atoms, and those are our trirose monosaccharides, again with 3 carbons. You can also have simple sugars with 5 carbons, those are called pentose. And then you can have hextrose sugars with 6 carbons. And the 6 carbon sugars are the ones that you want to commit to memory. Those are your glucose, fructose, and galactose. So make sure you know those 3 for your monosaccharides. For your disaccharides, these are when you have 2 monosaccharides stuck together to make a polymer. These are formed through glycosidic linkages of, again, 2 monosaccharides. And examples are maltose, lactose, and sucrose. And you want to make sure you know those 3. And make sure that when you're looking at the formula that you notice that they always have, or carbohydrates always have the elements of carbon, hydrogen, and oxygen in them. Okay, carbon, hydrogen, and oxygen. This next slide is just showing you a little bit of comparison of our monosaccharides and our disaccharides. Again, our monosaccharides are on the left-hand side, and the 3 that you want to remember or commit to memory is glucose, fructose, and galactose. Remember, these are simple sugars. Your disaccharides are 2 monosaccharides stuck together, and the 3 that you want to remember are lactose, maltose, and sucrose. Okay, disaccharides, 2 stuck together. Last classification category that we need to be aware of for carbohydrates is polysaccharides, and these are formed through the linkage or glycosidic linkages of many monosaccharides. And examples of polysaccharides are starches in plants and glycogen, which is stored in the liver. Of course, we use the glycogen for energy in animals. If you look down here at the image, you can see that the polysaccharides form chains of the different monosaccharides together to form the polysaccharides. Here's a little review slide. So I would pause the video at this time and try to put each one in the category it belongs in, whether it's a monosaccharide, disaccharide, or polysaccharide, before you continue on in the video and find out the correct answer. Number 1, glycogen is C, a polysaccharide. Number 2, maltose is B, a disaccharide. Number 3, glucose is A monosaccharide. Number 4, galactose is A monosaccharide. And number 5, sucrose is B, a disaccharide. Alright, let's now talk about some properties of carbohydrates. Some carbohydrates have a reducing ability. Some carbohydrates have an active ketone or aldehyde functional group. that reduce other compounds. And this property can be used to measure these substances. And other carbohydrates are non-reducing carbohydrates and do not contain an active ketone or aldehyde group. And an example of a non-reducing carbohydrate is sucrose or table sugar. So why would this property necessarily be important to us in a laboratory setting? Well, this property can help us determine what type of carbohydrate we may have. It may help identify. So it helps when testing for chemical properties. Other properties would be if a carbohydrate is a stereoisomer, meaning it's the same order and types of bonds, but in different arrangements and properties, meaning the structure is a mirror image of each other. Most sugars in the body are in the D-isomer or dextrose form, meaning they're clockwise in a polarized light plane. And you can see this is an example of D-fructose. But there's also what's known as an L, which stands for levo, or levorotary, meaning counterclockwise, and this is in a plane polarized light of L-fructose, which moves in the opposite direction of D-fructose, and you can see that example here on the right hand side. And there's some more examples in your textbook on page 178. So here's 3 questions that we just covered in the material. So pause the video at this time and try to answer these 3 questions before you continue on and get the answers. The answer to the first question is, name a carbohydrate that is a non-reducing sugar. The answer is sucrose or table sugar. The second answer would be define a stereoisomer. The answer is mirror image or same order and bonds, but arrangement is different. The third question is, most sugars in the body are in the D or dextrose isomer form. This next slide is just some terms you need to be aware of in relationship to carbohydrates. The first term is glycogenesis. This is the conversion of glucose to glycogen when cells' energy requirements are being met. This usually happens in the liver mainly, but it can also happen in the muscles. Next is glycogenolysis. This is the breakdown of glycogen from glucose. So anytime you see lysis, think of breakdown of something. Next is gluconeogenesis. This is the formation of glucose from non-carbohydrate sources. So neo, when you see neo in a word, think of something new happening. This happens in the liver and the kidneys usually, and your non-carbohydrate sources can be lactate from the muscles, amino acids from your proteins, glycol that's found in the liver or in fat tissue. Next is glycolysis. Remember, lysis means breaking down of something. This is the conversion of glucose and other dextrose into lactose or pyruvate. Next is lipogenesis. This is the conversion of carbohydrates into fatty acids. And then finally is lipolysis. Again, lysis, breakdown, and it's the decomposition of fat. Carbohydrate metabolism. Glucose is our primary source of energy. Our brain and our nervous system must get glucose from extracellular fluid. It cannot store it, so a decrease in available glucose causes loss of body function. Enzymes such as amylase from the saliva and pancreas and maltose from the intestinal mucosa hydrolyze or break down by chemical reaction with water carbohydrates to monosaccharides for absorption, and they're transported to the liver. Only glucose can be used directly for energy. So glucose equals energy for the body. So there's different biochemical pathways to break down glucose for energy or ATP. Again, glucose is the primary source of the body's energy and is broken down in phases in the body's cells. Last semester, we learned about glycolysis and the EMF pathway, or the Ebden-Meyerhof pathway, but now we're going to learn a couple more ways in which the body can gain ATP or energy. So we already know about glycolysis, or the EMF pathway, but you can also get ATP through the citric acid cycle, or the Krebs cycle, and the electron transport chain. Okay, so key points you need to know about the breakdown of one molecule of glucose for ATP during the Ebden-Meyerhof pathway, or the glycolysis pathway. Glycolysis of glucose is going to change into pyruvate or lactate, and this can occur with or without oxygen. Okay. Glucose consists of six carbons and is broken down into 3 carbon molecules. And the process of glycolysis takes 2 ATP in the beginning, makes 4, and you end up with a net gain of 2 ATP. That's what we learned last semester. And that's pretty much as far as we went as far as learning about energy and ATP. But you can also get energy or ATP from the citric acid cycle, or the Krebs cycle. This process occurs in the mitochondria with oxygen, and it generates 2 more molecules of ATP. Again, we're breaking down glucose. Next, during the electron transport chain, you can have many ATP generated. You can have somewhere in between 32 and 34 more ATP molecules generated for energy. The entire process for one molecule of glucose being broken down, the cell can get an average of 36 molecules of ATP. Okay, so the body has some different options to get the energy that it needs, or the ATP that it needs. The body can do glycolysis, it can do the citric acid or Krebs cycle, or we can do the electron transport chain. Again, these are all ways the body breaks down glucose for energy. This is just an illustration of the 3 different processes of getting our energy, or our ATP. Glycolysis, you're going to end up with your 2 net gain, 2 ATP. The center part is our citric acid cycle, and you're going to get another 2 ATP. And the very bottom portion down here is our electron transport chain, and you can get up to 32 to 34 molecules of ATP from our breakdown of glucose. Okay, the body needs a way to regulate carbohydrate metabolism, and there's hormones that work together to keep glucose at normal ranges, and this usually happens in the liver, the pancreas, and other endocrine glands. And one of the hormones is insulin, and it's produced in the beta cells in the islets of Langerhans. This is a group of cells in the pancreas. It's released when there's an increase in body glucose. It causes movement of glucose into cells and increases glucose metabolism. It's called a hypoglycemic agent, which lowers plasma glucose levels. Then there's glucagon, which is produced in the alpha cells in the islets of Langerhans, and it's released during stress and fasting, and is a hyperglycemic agent, and it stimulates glycogenolysis, gluconeogenesis, and lipolysis. Then we have epinephrine, which is produced by the adrenal medulla, which is located inside the adrenal gland, which is located on top of the kidney, and it inhibits insulin secretion, increasing plasma glucose levels. and it's released during times of stress, and this is called the fight or flight hormone. We're continuing on with hormones that help with the regulation of carbohydrate metabolism. Next, we have glucocorticoids. Cortisol produced in the adrenal cortex in response to the hormone ACTH increases plasma glucose and is an insulin antagonist. The adrenal cortex is the location is the outside portion of the adrenal gland, which is also on the top of the kidney. ACTH stands for adrenocorticotropic hormone, and the pituitary is a gland located in the brain. Next, we have ACTH and growth hormone. It's produced by the pituitary and it increases plasma glucose, and this is also an insulin antagonist. Then we have T4 and T3, and it's produced by the thyroid and it increases plasma glucose with minimal impact by increasing glycogenolysis and gluconeogenesis. And then finally, somatostatin, and it's produced in the delta cells in the islets of Langerhans, and it inhibits the secretion of glucagon and insulin, and it does not appear to have direct impact on glucose. Here's another trivia question to test what you've learned so far. Pause the video at this time and try to answer it on your own before you see the answer. And the answer is the conversion of glucose and other dextrose into lactate or pyruvate is D, glycolysis. This is another opportunity to try to review what you've learned so far about the hormones. You're going to try to match the description with the name of the hormone. Again, pause the video at this time. Try to do it on your own before you get the correct answer. Number 1, insulin is C. Number 2, glucagon is D. Number 3, epinephrine is A. Number 4, cortisol is E. Number 5, growth hormone is B. Next, we're going to talk about diabetes mellitus, or DM. This is when patients have hypoglycemia or high sugar. So let's break down the term hypoglycemia. Hyper means increase. Glyco means glucose. Anemia means dealing with the blood. So you have increase in glucose in the blood. You have 4 main categories of diabetes mellitus. You have type 1, you have type 2, you have other specific types, and then you have gestational diabetes. Let's first talk about type 1 diabetes mellitus. It's usually onset in childhood or adolescence. It's an autoimmune destruction of the beta cells. It used to be called IDDM, or insulin-dependent diabetes mellitus, and it makes up about 5 to 10% of diabetics. There usually is some kind of initiating event, such as a viral infection or an environmental factor that leads to the production of an autoantibody in a genetically predispositioned person that causes the onset of type 1. There's usually an abrupt onset, there's insulin dependence, and there's ketosis in these patients. Ketosis is a process where the body doesn't have enough carbs to burn for energy. And symptoms include polydipsia, meaning these patients are usually thirsty, polyphagia, excessive eating but not satisfied, and polyuria. These are known as the 3 Ps. There's also usually rapid weight loss, hyperventilation, mental confusion, microvascular problems, are all symptoms and conditions seen in diabetics. For type 1 diabetic mellitus patients, they're more likely to develop diabetic ketoacidosis, which is where the body produces too much or excess blood acids or ketones. Normally, our insulin allows glucose from the blood plasma to enter the cells and be broken down for energy or ATP. But in diabetics, no insulin, thus glucose can enter, and thus there's an increase in the blood plasma. Thus, the next resort, the body breaks down fats for energy. However, the intermediate breakdown products of fat metabolism are the ketone bodies. Thus, the patient's blood pH becomes too acidic, which can be deadly, as our blood pH must be between 7.35 and 7.45 in order for our enzymes to act as catalysts for our body's chemical reactions. Therefore, diabetic ketoacidosis can be a life-threatening condition. Here's just another trivia question. Which of the following is a characteristic of type 1 diabetics? Again, pause the video at this time. Try to answer it on your own before you move on to get the answer. The answer is all of the above. Now let's talk about type 2 diabetes mellitus. It's an insulin resistance or insulin secretory defect. It's usually onset in adulthood, and it's usually a result of obesity or an increase in fat around the abdomen. And it's also related to lack of physical exercise. And it's the most common type of diabetes. And it's usually a milder form. And it accounts for about 90 to 95% of diabetics. And it's usually referred to as a non-insulin dependent form, and cells become insulin resistant. People usually have a strong genetic predisposition to become type 2 diabetics. There's usually no autoantibodies involved in type 2, and usually for type 2 diabetics, women are usually more prone, also blacks and Native Americans. Here's our last question for this part of the lecture. Which form of diabetes usually manifests itself later in life and is associated with obesity, physical inactivity, and increased glucose in blood and urine? Pause this and answer it on your own before you go on and get the right answer in the video. The correct answer is C, type 2. This concludes part 1 of our carbohydrates lecture. I will see you over in part 2. If you have any questions, feel free to email me or see me during lab time.